And take it away, Jordie.
Sorry. Oh wait. There. All right. Okay.
Let me just double check. Did people get the recording, the little message that it's being recorded?
Yes.
Okay, good.
Yes,
Oh,
yes, got it.
I
okay,
got it.
found it. I just want to make sure we didn't do all this and then not have a recording. Okay, so we'll start from here. Go, Jordie.
Hi, my name is Jordie Ho. I am a PhD student in the Department of Plant and Environmental Protection Sciences in the College of Tropical Agriculture and Human Resources at the University of Hawaii at Manoa. Today, I'll be talking to you about my work in ecology, evolution, and emergence of plant pathogenic viruses with a focus on Higrovirus waimanalo. So Higrovirus waimanalo is a plant pathogenic virus of the family Chaetaviridae. It was first described here in Hawaii, infecting different citrus species, Hibiscus arnottianus or Kokiokeokeo, Hibiscus tilaceus, and or how. So Kokiokeokeo is one of our native Hawaiian hibiscus. It has a nice fragrance, and you'll see it all across the UH campus. And actually, on some of the islands, subspecies of Kokiokeokeo are endangered. And Hau is thought to be one of our canoe plants. So what's unique about Higrovirus waimanalo is that it's vectored by mites. And there's only about two other families of mites that vector plant viruses, and most plant viruses are vectored by insects. Mites aren't insects, they're more closely related to spiders. And what's really interesting is that each of these spots that you see on this leaf are where the mite fed. So this particular virus is actually thought to be more of a mite infecting virus than an actual plant virus. So when I say that, I mean that it's sort of like a cross kingdom thing. Like how coronavirus infects animals and animals don't have symptoms, the mites also don't have any side effects from being infected with this virus. But when it jumps a host, like to a plant or with coronavirus to humans, then we get a pandemic because humans aren't immune to that or need to build up an immunity. And so that's why investigating this is really a unique system. And it's also important because Higrovirus waimanalo is related to a really destructive pathogen called Silovirus leprosus. It's mainly in Central to South America, and so far it hasn't moved northwards towards North America, but it has cost the Brazilians about $55 million are spent on pesticides to control the mite that spreads it. 9.5 million boxes of oranges were lost due to aesthetic value, or the virus also causes premature fruit drop. And so you can lose oranges this way. And about 9.5 billion boxes of oranges is worth $50 million in profit. But Brazil still remains to be one of the top producers of citrus and oranges in the world, although they've seen a decline due to not specifically this pest, but a compounding effect of this pathogen as well as others. And these are just the symptoms across the top. It causes these ring spots on stems, leaves, and fruits. And in severe cases, the tree can die, which is, again, contributes to the loss of produce and production. So this Higrovirus waimanalo is a non-systemic plant virus. And what that means is that if you see the symptom over here, and you see those ring spots, you won't find it over here. So it's actually just in one location. And if you pull this leaf off and you don't have any virulliferous mites anywhere, then your plant is pretty much clean, which is in contrast to some systemic plant viruses, which are the most common plant viruses, where you'll find the plant viruses everywhere. So you'll find it throughout this whole bush. What's interesting is that, so plant viruses are unique in that they have these proteins called movement proteins, and that's how you know it's a plant virus, because plants have cell walls and the viruses can't get through the cell walls because of the little opening called plasmodesmata. This isn't to scale, but the virus particles and most virus particles are too big to move through plasmodesmata. So these movement proteins help them move through by either opening the size or increasing the size of the plasmodesmata so the virus whole particles can move through to the next cell and then into the phloem for long distance transport. Or they can modify different structures in the plasmodesmata and move smaller portions of the virus, like virus particles or things that direct virus synthesis through the plasmodesmata, and then into the phloem and throughout the whole plant. What's interesting is that Hegrovirus wymonol actually has these movement proteins, but they don't seem to be functioning, which is possibly because they haven't overcome plant host defenses, or they just acquired the wrong type of movement protein. And that's what I'm looking at. Like, why isn't this plant virus able to move? To do this, I'm working with a clone of the virus. Basically, it's a synthesized version of the virus with the whole genome in 3 different plasmids, which are just genetic constructs of the virus. And what I'm doing is I'm working on right now attaching a fluorescent protein to the movement protein so I can see where the movement proteins are. Where are they localizing? Why aren't they moving through the plasmodesmata? And so I have to do different trials because you have to work with the different protein structures and adding this fluorescent protein. And so I've been working with common bean plant. It's common practice to try it with this first because beans are easy to grow, they're fast, you can grow a lot of them quickly. And so basically I prepare the virus in a mixture, and then I'll use a syringe to to force the mixture into the leaves. And so that's day 0 where I freshly inoculated them. And then there's day 7. So day 7 under regular light and day 7 under a UV lamp. And it might be misleading because the UV lamp makes it look like it's glowing, but that's actually just the damage from me inoculating it. So each lesion, that's where the virus is, but it's not necessarily glowing. It's just the different contrast. And so once I get this to work, I'm gonna go to the laser scanning microscope in the bioelectron microscope facility that we have on campus, and hopefully I can see where the proteins are located in each of the cells, as well as determine where they're being stopped. And so these are my expected outcomes. Refining this tool that I already have from a previous PhD student to investigate the non-systemic viruses, contributing to knowing where the virus particles are localizing in the cells. So know, like, is it stuck here, or where is it stuck? Why can't it move intercellularly? As well as looking at control or interfering with systemic viruses. So we know why non-systemic viruses move. We can figure out possibly how to stop or block systemic viruses from moving around. Because once you get a systemic virus, you usually have to get rid of your plant, and it's sad. And so this is a summary. My project provides looking at a unique pathosystem, which is non-systemic plant viruses vectored by mites. It provides fundamental research by looking at the evolutionary step between plant viruses and an arthropod virus, which leads to the emerging of new plant viruses. Multiple hosts are naturally susceptible, which is important because if they're close together, you can transfer the virus to wherever, as long as they're susceptible. And it provides a look at applied research, looking at non-systemic viruses that have a polyphagous vector. Breviplpus mites are found on many different plants, from tomatoes to ginger, flowering ginger. And they also threaten an endemic and endangered species, which is Kukio, our native Hawaiian hibiscus. And it represents emerging pathogens, which could spread and cause problems to our hibiscus plants. I'd like to acknowledge my advisor, Dr. Michael Melzer, as well as current and former members of the Agro-Security Lab, the Pacific Continental Network for funding my GA ship. And if there are any questions.
Suzanne, did you have any questions for Jordie?
Sure, that was very interesting, Jordie. Thank you very much for that work. I guess I was thinking of two questions. One is what was the most challenging part? What has been the most challenging part of your research to date, and how have you overcome it? And
Oh, mhm.
well, let me pause there. Yeah.
Oh, okay. Okay. Well, the most challenging part is figuring out where to place the fluorescent protein into the virus, because viruses, I only have the sequence and I don't really know how they're folded or what's going to happen once I place the fluorescent protein in there. I mean, like, I know if it works, it'll light up, but I know if it's not working, it won't light up. And I won't really know why. So it's a lot of just troubleshooting, making sure that I have the exact sequence of the virus so that I can correctly insert it. And that's my main problem. And right now I'm overcoming it just by sort of just keep on trying, just trying different places of inserting it, looking at different protein folding programs so I can figure out where there might be a loop to insert the fluorescent protein, because you want it to not interfere with the structure of the movement protein. So fluorescent proteins are really big, and you want to make sure that that big structure isn't getting in the way of what the regular movement protein is functioning or how it's functioning. So that'll be the next step, just making sure that it's still working or not working. I guess the movement proteins don't work. So yeah.
Very interesting. And if I could ask my other question, it sounds like you've got here an example of the virus infecting an endangered plant, an endemic plant that's endangered, that is in some cases endangered, a canoe plant, and then examples from Brazil, at least, of it infecting agriculture. So would you say that this is a virus that has very broad potential concern in Hawaii across all of those um, areas?
Yes. So in plant pathology, we have this thing called the disease triangle. And what you need to make the triangle is you need a susceptible host, you need the right environment, and you need a virulent pathogen. So with climate changing and warming, this might provide a better environment for these viruses that I'm talking about, like citrus leprosis or sillovirus leprosis, as well as Hegrovirus Waimanalo, to spread further into different, maybe, so hibiscus belong to the family Malvaceae, so different Hawaiian endemic Malvaceae that are endangered. And so it is concerning that these might move, or they also might pick up a mutation, which allows them to further expand their host range. And so I do think it's an important thing that we have to survey for, or keep looking for, or monitor,